He's also currently researched acute and chronic gut disease, specific projects on pro and prebiotics, gas production, gut microbiota development with age, gastroenteritis, obesity, and colonic homeostasis are being also carried out. Human trials are a major facet on this research, with initial principles being tested in gut models. He has had over 140 research contracts. He has published 8 books, more than 490 research papers, and has graduated over 75 PhD students, some of them co-supervised. His age factor, it's 96 according to the Web of Science. He's also listed as a highly cited author by Clarivate, and his PhD was on bacteriology of sediments and was awarded by University of Dundee. He then had research positions at MRC Dunn in Cambridge and the Institute of Food Research in Reading. So we, as you can see, we are very we're glad to have this kind of high-level speakers today. Thank you so much, Professor Gibson, for being with us. So now, before we start, please remember to mute your microphone during the presentation. And if you have any questions through it, you can either write them on the chat so that you do not forget them. And at the end, you can ask them directly to our speaker, or I can read them aloud for you. Welcome. And again, thank you so much, Professor Gibson.
Okay. Thank you. Hope you can see my screen now, yeah?
Not yet.
Not? Okay. I think, Friedana,
I was sharing.
I think you need to switch yours off to allow mine to control, I think.
It's already off.
It's there now? Great.
No, could you please again try to share it? Mine is already off, so it shouldn't be.
Okay, let's try that again. Okay. Share screen. Let's try that again. This time I think we're okay.
Now it's ready. Yeah. Thank you so much.
All right. Sorry for the slight delay, everyone. Thanks to Friedana for the very nice introduction, and obviously the invitation, and which was done through my friend Niranjan, who I'm sure many of you know. He and I have worked together for a long time, and he's a great person as well as a great scientist. So thank you, Niranjan. Thank you for the invitation. And my area is human gut microbiology. As you heard, my background is actually in the microbiology of sediments, but I've worked in the human gut area for 30 years. The sediments was part of my PhD study. And what I did in my PhD was actually look at the production of a gas called hydrogen sulfide in sediments. This was having it's a bad smelling gas. It was having quite a noxious effect in the environment of the sediments. These were sea lakes and estuaries in Scotland, and H2S was being made by bacteria in these sediments. I then kind of moved to Cambridge in the United Kingdom and maintained my interest in the same bacteria, but this time in the human gut. And I began to look at a disease called ulcerative colitis in regards to H2S production. But it became kind of apparent that most bacteria in the human gut were actually not pathogenic, were commensal or even positive for health. And I moved my research focus away from the bad news, if you like, into the more good news, and begun to look at probiotics and prebiotics. And I'll define, if you don't know what they are, don't worry about it, because I'll define those as we move through the seminar over the next 45 minutes or so. So I'm going to do this as an A to Z guide, and I guess one of the good things about that approach is that you will know when we are getting to the end, because once we get down to STUV, we're on the kind of downward slope. But before I start, this is the first slide here. This is the department Niranjan and I work in on the left there, which is Department of Food and Nutritional Sciences. University of Reading, which is about 25 miles west of London in England. And we kind of have a long tradition of food bioengineering run by Niranjan and other people who have looked at all sorts of different physicochemical aspects of food, including their structure, their matrices, sensory qualities, enzymology, food quality, and so on. We also have a group who are interested in nutrition and do human intervention studies to look at antioxidants, cognitive function, polyphenols, immune function and cardiovascular health. And then we have the group I'm largely affiliated with, which is a microbiology group. We have some interest in food safety, and we also have some interest in gut microbiome, which is what I'll be talking to you about today. So that's the department on the left. On the right there is really what I work on and have worked on, I'm afraid, for the last 30 years, because that is a picture of human feces. This is a scanning electron micrograph of human feces with, you can see there lots of little squiggly shapes and entities in there. Most of what is on that image is bacteria, rod-shaped microbes there, cockle-shaped ones, coccobacilli, and actually about 70% of the dry weight of human feces is microorganism. The main constituent, of course, of human feces is water, but if you just look at the dry weight basis, something like 70% is microbe. So they're going to have a pretty profound impact upon our physiology because there are lots and lots of them there. And then in the middle, I don't want to gross you out too much, but that is a virus. We all know that viruses have been in the news for more than a year now in a massive way, and will be for the foreseeable future. Viruses do occur in the gut, and this is a picture of a virus from the gut. And what you've got there is the head of the virus, the phage, and then its tail there. And as I said, I don't want to gross you out too much, but I'm happy to admit that this is a picture of viruses from my own fecal samples. I'm not sure what time of day it is where you were listening there, but if it's near lunchtime or whatever, I do apologize. So let's move on. Okay, these are three pretty famous pioneers in the gut microbiome area, Hippocrates, Arbuthnot Lane and Mechnikov. So the quote at the top in red, "Let food be thy medicine, medicine be thy food," has been famously attributed to the father of modern medicine, Hippocrates. But actually there was a research paper published a couple of years ago by Susan Prescott in Australia, who had researched this and actually suggested that Hippocrates did not say this famous phrase. But what it is suggested Hippocrates did say is all disease begins in the gut. Now, I'm not sure whether all disease begins in the gut, but certainly a lot of them do, as I hope you'll see in a minute or two. On the right there, Arbuthnot Lane was a Scottish physician who lived from 1856 to 1943. And he was notorious, actually, for saying that cognitive issues like migraine and schizophrenia had their roots, had their cause in the gut. And he suggested that toxins which were made by the human gut could cause schizophrenia and migraine. And actually, nobody really believed this. And Arbuthnot Lane passed away and really wasn't given the credit he deserved for those observations, because where we are now today is that it does seem there is a big link in between the gut and the brain. And indeed, certain chemical neurotransmitters which are manufactured by the gut microbiome, either positively or negatively, can transmit to the brain and affect a whole range, a whole cascade of cognitive issues, from depression through autism right through to dementias. And clearly there is a lot of interest in trying to modulate that interaction. And then at the bottom, the person in that picture is called Mechnikov. And he in 1907 wrote a thesis called The Prolongation of Life. And this thesis was really driven by people who lived in Bulgaria, and Mechnikov observed that Bulgarians had long, happy, healthy lives. And he looked at their diet and he suggested that their intake of what he called sour milk, so this was milk with bacteria in, was helping their happy, healthy lives. These bugs in the milk were getting into the gut and having a good positive effect upon gut health. And this was the birth of really what we now call today a probiotic. So the intake, deliberate ingestion of positive microorganisms is the probiotic concept, and that started with Mechnikov's thesis in 1907. And he was given the Nobel Prize for those observations. He worked at the Pasteur Institute in France. I just wanted to do three definitions for you before we get into the A to Z. The first one is probiotics, which, as I already mentioned, are live microbes and bioactives. they've been around for a long time in human civilization, thousands, if not hundreds of years. But they're now today as popular as they have ever been. And in the United Kingdom alone, it's estimated that about 20 to 25% of households take probiotics on a daily basis. Prebiotics is something which we do most of our research on in the gut area at Reading, but it's something which is newer than probiotics and is defined like that. So prebiotics are more like the fertilizers for the microbiome. So these are dietary ingredients which pick out components of the indigenous flora which are positive for health. So probiotic is the live microbes in the diet. A prebiotic is something which grows the ones that are already in there in the first place. And then the next slide is, the next definition is that of a synbiotic, which is simply just a mixture of the two, adding a probiotic and a prebiotic together. So those are the three things which we do our research on. If you want more information on this, I'd direct you to the website of ISAP, ISAPP, which is International Scientific Association for Pro and Prebiotics, where you would find lots of nice infographics, videos, research papers, and a whole host of other information on what pro, pre, and symbiotics are all about. So let's go ahead with the A to Z now. So I've picked out for each of these letters something that I hope is relevant to the human gut microbiome and gives me the opportunity of showing you some results, as well as some kind of brief stories behind each of these approaches. And the first one I've picked on is autism. As I said earlier, there is a strong connection has been realized between the gut and the brain. And we've been interested in autism since I think about 2007, something like that was our first efforts at researching this condition. And autism is, of course, a spectrum of different disorders, which begins to get identified at the age of about 4 to 5 years of age. Now, there is an observation that children in particular who have autistic spectrum disorders suffer lots of gut difficulties. They complain of gastroenteritis frequently, of abdominal discomfort, lots of gas, distension, and pain. And this led us to look at the microbiology of their gut. And what we found was that microbes called Clostridia were in prevalent numbers in the GI contents of 50 children with autism in comparison to children who did not have autism. And Clostridia are generally seen to be quite negative for health. And so whatever the reason for this predominance of Clostridia in the autistic situation is, this is not really doing the kids any good and may well explain difficulties that they have with their gut. So we've done various trials, we've done probiotic trials, but the more recent one, published a couple of years ago, was a trial with the prebiotic, which is, as I said, a boost for positive bacteria. And what we were trying to do was boost the beneficial ones at the expense of the Clostridia. And so we did that with a prebiotic called GOS, or galactoligosaccharide. Human intervention study was carried out. We looked at microbial composition in feces. We looked at metabolite profiles in urine and feces through NMR. And then we asked the volunteers, the children or the carers or the parents to fill out subjective questionnaires on symptoms, moods, and behavior. What we saw when the prebiotic was fed was an improvement in their gastrointestinal microflora. Bifidobacteria were increased. Metabolite profiles in urine and feces were changed. Predominantly acetate was more prevalent, and improvements in symptomology also occurred. And one interesting thing, which was kind of an accident in the trial, was that we asked about sleep patterns as part of the questionnaires. And this was something that had kind of crept in fairness, from another study and wasn't really the main remit of this autism trial. But what did occur was when the improvements in the gut contents occurred, the children reported better sleep patterns and better duration of sleep. And at this stage, we have no idea why that has occurred, but it's something we'd like to take forward in our research in the future to understand what the relationship between modifying a person's gut microbiome and sleep patterns may be. Coincidental to that, a BBC program called The Truth About Sleep, on the bottom left there, the presenter of this is an insomniac, and he decided to take an intervention for the gut to see if that would help him. And coincidentally, that was the same intervention as we tested in our autism trial, and he improved his sleep ratios as well. So we don't know what the link is, but it's something that we will look forward towards in the future. B is for Bifidobacteria. These are seen as the good guys. These are popular probiotics along with lactobacilli. They're called Bifidobacteria because they bifurcate when they replicate like that. They show different prevalence ratios during life, in that during the early life, they're quite high, particularly during breastfeeding. And then when we move from weaning into adult years, they decrease. The blue sector is Bifidobacteria. And in particular, as we age, they are at a pretty low level. So these are high in early life and low in later life. And as I said, these are seen to be very positive for gastrointestinal welfare. And just to amplify that, the bar chart there on the top right shows you results from a trial published some time ago now in children who were formula fed. Now, breastfed kids have high Bifidobacteria, formula fed a little bit lower. And what the trialists did here was add to formula a prebiotic to try and replicate the bifidogenic effects of breastfeeding. And what they did was measure atopic issues, so allergic outcomes in response to the addition of the prebiotics to the formula. So what you've got there in maroon is the control. So these are infant formula without prebiotics. And then in green, the same formula with 2 prebiotics added to them, which are called GOS and FOS. And right across the board there, you see reductions in atopic issues. But the important thing that impressed me about this trial was that these data are 5 years after the intervention was carried out. So the kids were fed the formula for the first few months of their life, but what the trialists did was monitor outbreaks of allergies for 6 months, 18 months, 24 months, and 5 years. And 5 years later, as you can see, generally, the atopic issues had been dampened down, comparing the green to the maroon there. C is for colon. Most of the bacteria inside the large intestine, inside the gut, are inside the large intestine. On the right there, you've got a cartoon or a picture of different GI tract contents: the stomach, the small intestine, the large intestine. It doesn't really matter too much about the bacterial names, but what we can see is that numbers of bacteria do differ depending on which organ we are considering. The stomach has pretty low populations. The small intestine are a bit higher, 10 to the 6 there, 10 to the 3 up there, and at the end, the top of duodenum, and then at the ileum, a bit higher, but in the large intestine, higher again, 10 to the 11 to 10 to the 12 colony forming units per mil in every inside the large intestine. So the numbers predominate inside the colon. And the reason for that is that pH is favorable more so than the stomach or small intestine, as is transit time, because the transit time of a person's large intestine is on average in an adult about 2 days, but the transit time of the stomach and small intestine is just 30 minutes and 2 or 3 hours respectively. So 2 to 3 days inside the large intestine is a long time for bacteria to grow up to very high levels indeed, and that is indeed is what happens. So most of the microbiological action inside the gut is in the colon, and that's why I picked C for colon. On the left there, you've got an opened up view of a real large intestine, and this is from a sudden death victim. It's a pretty awful picture, as you can see, but this was somebody who had a sudden fatality and was perfectly healthy up until the point of their death. And within 2 hours of that fatality, we took their large intestine at autopsy, and we measured the bacteria which were present in different regions of the colon, the substrates they metabolized, the products they made, as well as the enzymes they had, and used that to build models of the large intestine, which we operate in our labs in Reading today. And so we know that these are valid models for microbiological analyses that get very close to what happens inside the real gut. And this has been pretty good because it's obviated the need for us to do any animal research in our capacity, because we know we can get closer to what happens inside the real human large intestine with our gut models. And the gut models, I'll show you a picture later on, are just basically fermenters linked together by a series of tubing with filters, pumps, growth media, and inoculated with fecal bacteria. I'll show you a picture later on. But what we do is use these to test interventions before then moving into human trials. In the triangle there on the top left, those are kind of factors which can impact upon the composition of the large intestinal microbiota. Now, we usually live in harmony with our microflora, but those are kind of challenges which can upset the relationship and predispose towards clinical states if they become too prevalent. D is for diarrhea. This is a very, very common complaint. I'm sure most of you have experienced diarrhea, probably from gastroenteritis, food poisoning, if you want to call it that. It's something which kind of gets probably every one of us at least once in our lives. And if you had food poisoning, I'm sure you remember being pretty ill for a few days and not really being bothered to do anything. And the body's reaction, of course, to food poisoning, gastroenteritis, is to produce diarrhea to try and get rid of the pathogen. And we really just have to put up with it until the thing sees its course. The bottom right shows you the results of a volunteer study from us a few years ago, where we measured rates of gastroenteritis in people who were going on holiday for high risk incidence of GI tract infections. In maroon, you've got a prebiotic group, and in blue, you've got a placebo. So 2 weeks before the people went on vacation, half of them were given a prebiotic, and then they took it while they were away, and the other half were given a placebo. So as I mentioned, the prebiotic is meant to stimulate beneficial bacteria inside the gut. So what happened in the prebiotic study, prebiotic arm of the study, as you can see, was a reduction in incidence of diarrhea, a reduction in its duration, and a non-significant reduction in abdominal pain. So this suggested to us that positive bacteria, Bifidobacteria, had grown up in response to the prebiotic and had a concomitant effect upon gut pathogens, which otherwise would have caused issues. And if you look at the map of the world there, those are areas of the world where for travelers to those places, gastroenteritis becomes a bit of an issue. And to this end, we worked a few years ago with the Olympic Games team when the Olympic Games was held in Rio in 2016 in Brazil. And when British people travel, they're not very robust with their guts. And so the Team GB athletes took a prebiotic to try and minimize their chances of infection before competition and during training. And all of the trialists, all of the Olympians in the Team GB took it. It was meant to just be the cyclists, but in fact, all of them did in the end. And when we look back on this, we might remember that Team GB were actually second in the Olympic Games and did very well. It was one of the best Olympic game performances Team GB ever had. And I would be remiss of me not to say that the University of Reading takes the full credit for that. I can't believe I just said that, but yeah. Anyway, if we played a little role, we'll be pretty happy. And it's a secret, so don't tell anyone. When the Tokyo Games happen, they are going to take it again. And this was a prebiotic which we made with the expertise of people like Naranjan using our food processing facilities at Reading. So it was something which was researched on a bench in our lab, moved into the pilot plant, produced enough for a volunteer trial, and one of the trials was the diarrhea study there. On the top left there, you've got an advert from British Airways magazine. So this is British Airways in Flight magazine. And this was a product which was made as a result of the studies which I've just mentioned, and which British Airways advertised as supporting your tummy while abroad. And in the small print there, you can't see it. It's far too little to read, but it says based on research carried out at the University of Reading. And that is my kind of claim to fame, if you like, because I can't get my research in high-powered journals like Science or Nature or PNAS or anything like that, but I can get it into British Airways High Life magazine. I'm sure they have a very rigorous refereeing procedure. EFSA is E. This is the European Food Safety Authority. EFSA have been approached about claims, and their responsibility is to try and identify whether claims on things like prebiotics and probiotics can be justified. And it hasn't gone particularly well, because the vast majority, if not all, of the claims which have been submitted in this regard have not made the grade. And so as a result of that, we've seen in the past newspaper headlines like these, which is all kind of quite obviously bad news and these things don't work, etc., which to me is contradictory to the science, because at the moment where we are now is that there are 26,000 plus research articles on probiotics and about 5,000 or 6,000 on prebiotics. So that's well over 30,000 research articles in total. And I'm sure that in there somewhere, there must be a series of claims which can be made on health, because good techniques are used in the field, the trials are well done, they're controlled, they're published in decent journals, etc. But we're not making the next step in terms of getting the message across appropriately. And with the best will in the world, some probiotic products are better than others. Some do very well and have been well tested independently, and some have no science behind them whatsoever. And one way or another, I think we need to unravel the wheat from the chaff and begin to really get to grips with this legislative picture, such that people are better informed about which products can do what. And I feel that in areas of the world apart from Europe, things are progressing much better. Health Canada has a very robust legislative and clear legislative process. The USA similarly, and certainly Asia are very used to having health claims verified or otherwise, depending on their science. So it's kind of a little bit concerning to me, this hard stance that occurs in Europe at the moment. G, I've missed a letter. Let's go there. F, sorry. I'm not very good at my alphabet, am I? So F is for fast facts about gut bacteria. There's just some facts. Our body has 10 times more gut microbes than human cells. So you and me are 90% microbe and only 10% human. We excrete the same weight as 12 elephants in our lives. The gut is the microbiota is the most active organ in the body because of the sheer weight of numbers of microbes. I read a couple of months ago on the right there that in humans, it's suggested that 12 organs can be influenced by the gut microbiome. So we're talking about a big impact upon health here. On the left at the bottom, about 3/4 of our immune system resides in the gut. This is called GALT, gastrointestinal associated lymphoid tissue. Bacteria are antigenic because of their cell wall. And if you've got 90% of the cells in your body being microbes, that's going to have a massive effect upon immunity. And on the bottom right, there are over 1,000 different species of bacteria recognized in the human gut now. Let's go to G. This is the interactions of the gut. You can see, you can read them for yourself there. These are all health-driven interactions which arise from the gut. And things that you would expect to see on a slide like this are present, such as pathogen inhibition, IBD, IBS. This is inflammatory bowel disease, irritable bowel syndrome, gut disorders you would expect to be mentioned here in terms of the interactions between the microbiome and the gut. What is more compelling potentially is things which are outside of the gut, which are on this slide, like atopic issues. That means allergic issues like eczema and asthma, obesity. The gut microbiome world went a little bit mad a few years ago by linking obesity, or at least partially, into the gut microbiome. And then at the top there, cognitive interactions. If there truly is a link between the gut and the brain, it means that we can actually begin to modulate that activity, hopefully. H, I chose hospital infections. On the bottom left there, you can read the data. It's very UK driven, but that's the kind of cost of hospital infections just in the country I live in now. But I just wanted to point you towards the two bar charts here. These were two separate trials on probiotic use in hospitals with a view towards trying to control diarrhea, and specifically diarrhea caused by Clostridium difficile. So the top one here, in blue there, you've got outbreaks of diarrhea in people in hospital taking antibiotics, generally in the controlled population, about 18%. When a probiotic was taken, which is a lactobacillus, that was dampened to just 5%. And C. difficile went from 6% in the control down to 0 when the probiotic was taken. Same kind of thing occurred here. This was a trial in London hospitals in yellow, 34% of the patients experienced diarrhea in hospital. That was dampened to 12% in a probiotic population. And similarly, 17% experienced C. difficile toxin in feces. That was dampened to 0%. So these are very straightforward approaches whereby the incidence of hospital-acquired diarrhea can be markedly reduced. And I've said to anybody I know and have done for years who are going into hospital or are living in care centres or are taking lots of antibiotics, one way to potentially counteract some of the side effects is to take a probiotic before, during and after the antibiotic regime. I is IBS, irritable bowel syndrome. This is a very common gut problem particularly in Western populations. It's manifest by unpredictable bouts of diarrhea or constipation or alternating between the two. And hence IBS is given the title DCRA, standing for diarrhea, constipation or alternating form. And again, this was a result this time again of a prebiotic study. The bar chart shows you 2 different doses of GOS prebiotic, which were given to people with IBS in comparison to a placebo. And in green, you can see the rise there in Bifidobacteria when the GOS was taken. David Silk, a gastroenterologist, monitored symptomology in the IBS patients, and concomitant with that increase in Bifidobacteria were improvements in a range of symptoms of IBS evidenced there. Now, I said in my abstract, the hardest letters to fill in this alphabet were J and K, not X and Z, as you might guess. J I really struggled with, and K I struggled with. But in the end, for J, I came up with the name of a person, and this is Julie Elliott, and she is a member of Parliament, so she is a politician here in the UK. She actually comes from the same town in England that I come from, which is Sunderland. And Julie is the chair now of something called an APPG, which stands for All Party Parliamentary Group. Now, APPGs are groups of cross-party, so it doesn't matter about your political affiliation, so it's all parties to look at particular issues. And in the Houses of Parliament here in the UK, there are hundreds of APPGs. Every country in the world has one. Every sport you can think of has one, but there are very few science ones. And so we had the idea in 2019 to set up one, which was driven by the microbiome, and we now have an APPG which looks at the human microbiome and health. And some of the other people involved are at the bottom there: Kirsty Hunter, who's a nutritionist at Nottingham, Alan in the middle, Alan Barnard, who's a campaigner, and Gemma in the middle there, who works with us in Reading. Those are the people, together with myself and Julie, who set up this APPG. And the brief is summarised there in black. And what we are doing is trying to get key opinion leaders, members of Parliament, members of the House of Lords interested in the microbiome as a tool for health, and really objectively assess the science that is there, and have these people who have a lot of contact and a lot of influence translate messages if these messages are seen to be important enough. And this is something which it is something which I am really very positive about in the next few years, because it's okay for us, you know, in a way, to do our science and publish papers, but we need to get these communication messages across to people who can benefit from them. And the APPG, I hope, is an important route to that moving forward. So J was for Julie. K was another hard one. It's another person. K is Kelly Brook, who's I don't know what she is. She's either an actress or a TV presenter or something. But anyway, the reason I picked on her in a talk about gut microbiome is that Kelly advertises a probiotic bed mattress, and that's a picture of it. So this is a bed mattress with probiotic bacteria incorporated into it. On a similar vein, people are also now putting live probiotics into bathroom cleaners and disinfectants. And of course, probiotics have to be alive in these products to do any good. And probiotic in Greek actually means for life. So if you put probiotics into a disinfectant, I don't think they're going to survive very well. Similarly, I don't think they're going to do great in a bed mattress either. But anyway, Kelly says having a busy schedule, constantly on the go, don't always have time to look after my body in the way I would like. Having a bed with probiotic qualities means it's doing my body good while I sleep. You can't ask for more than that. Well, yes, you can. You can ask for probiotics to go in where they're meant to go, which is yogurts and fermented milks and drinks and so on. So anyway, I took the opportunity to write to these two manufacturers. I wrote to the bathroom cleaner, said, probiotics have to be alive. How the hell are they alive in your product? And they didn't reply. The bed mattress people, I said the same thing. How are they alive in your bed mattress? And what they did was refer me to their website. And that was what they saw. What I saw on their website was a picture of what the probiotics are meant to be doing in the bed mattress. So it immediately told me that no longer do we need to spend hundreds of thousands of pounds or euros doing volunteer trials, because we can just draw pictures and get our scientific validity. But what they suggested was that bed tights in the mattress were defecating, and that was feeding the probiotics, and the skin of the person on the bed mattress would also be a nutrient source for the probiotics. So anyway, yes, have a think about the probiotics wiggling around in your bed mattress when you go to bed tonight, but each to their own. L, we already mentioned Mechnikov Live Microbial Therapeutics, otherwise called probiotics, long use in humans, lots of research articles. Some of the things that have been looked at are diarrhea, irritable bowel, inflammatory bowel, cognitive health, allergic issues, infections, and colorectal cancer. But I just want to point that bar chart to you. This was a study which really caught my eye a few years ago with probiotics. It was a study from Kiel in Germany in respiratory infections. So this was carried out over the flu and cold season. And what the Kiel scientists did was feed a mixture of vitamins and minerals to a control group, and a mixture of minerals and vitamins to a test group, but with 3 probiotic bacteria added to the vitamins and minerals. And they then monitored rates of flu and cold. And what happened was there was vastly improved recovery from cold and flu in the probiotic trialists, and nearly 500 people took part in this study. So there's no effect upon incidence. The probiotic did not stop people getting flu, but what it did was speed up recovery by an average 2 days. The papers which were produced from the study also showed that there was 20% fewer symptoms when the probiotic was taken, and this was attributed to immune development because they measured the immune response in peripheral blood. So just bear that in mind. This is a study where probiotics are given, which go in the gut and have an effect on a respiratory issue. And I'll come back to that when we talk about COVID in a little bit. M is for museums. The last few years, we've been working quite a lot with museums. This is a walkthrough large intestine, which is at Winchester Museum, which we helped them build, which is about 30 miles from Reading. The London Science Museum a few years ago had a demo on appetite regulation called Cravings, and they built one of our gut models there. And this is the gut model that I mentioned at the start, which was validated against human sudden death victims. And this is my little touch of engineering, Naranjan, which he wanted me to say, and I hope I don't insult you. So this is as close as I ever get to real science and engineering. These are the models that we've built. There are 3 vessels, 1, 2, 3, aligned in series, which replicate the proximal, transverse, and distal area of the large intestine, fed with a growth medium through a pump there. They have increasing pH. This is slightly acidic. This is neutral. A circulating water bath pumps water around there in a jacket to keep them at 37 degrees C. pH monitors there regulate pH through electrodes there, and there to pump alkali or acid in as appropriate, and then they're all on flatbed stirrers, and the whole thing is anaerobic because it's pumped through with oxygen-free nitrogen. And these are ways in which we really test our interventions before moving on to human studies. And as I said earlier, we know that they do get close to the real human gut. So I'll speed up a little bit because we have to be finished, I think, in about 10 minutes, and then we'll try and do some questions. N is for non-intestinal. The microbiome is not just in the gut, it's elsewhere. It's in the oral cavity, the placenta, the vagina, the skin. People are even telling us, certainly the ear and the nose, People are even telling us now that the brain might even have a microbiome. But as I mentioned, the vast majority of the human microbiome is actually in the gut. Always for origin. This is a strange slide. I need a definition of feces for a talk a few years ago, and so I typed definition of feces into Google. And the first website which came up gave me a definition, which was great, and I got it and I used it for my talk. But I scrolled down the website and saw a map of the world. And this is a screenshot. I've not fixed this. This is a screenshot of the map of the world for the origin of feces, okay, from a website. And apparently the origin of feces was between 1425 and 1475. That opens up the question as what people did before then, but anyway, we'll park that. But look at where the origin of feces is meant to be, that red square there. If I'm not mistaken, that is 25 miles west of London, and that is right slap bang over the University of Reading, where Naranjan and I work. So I think that the origin of feces in 1425 was actually in Reading, and I think our students, when they graduate, should have T-shirts made which say, I work in the university where feces was invented. So there we are, and it's a screenshot, so it proves it. Prebiotic, I already said what they are. They're fertilizers defined like that. This shows you the variability of the internet. Between 3,000 and 5,000 peer-reviewed papers, depending where you look on the internet. The value is anything per annum between $3 and $16 billion. It's a huge range, again, depending where you look. Nevertheless, there is a rise in prebiotics, the types of products, new company developments, the research that is doing, and that's predicted to occur between 11 and 15% for the next 5 years. So that's for prebiotic. Q-Quorum sensing. Some of you who might have a microbiology background will know about quorum sensing. This is something which occurs in mixed microbial communities where the bugs basically talk to each other. They send messages to one another. Things like messenger RNA goes backwards and forwards. So quorum sensing goes on where interlocked microbial species can basically communicate in mixed culture like the gut. And this is just a bit of fun that we had a few years ago with archaeologists, where archaeologists gave us coprolites, and there's a picture of coprolite there, which are ancient fecal remains from 3 digs. One was a Roman dig in a place called Silchester, another one in Arizona, and another one in Turkey. And we got coprolites and we used DNA profiling to look at the microbial populations in ancient fecal remains to get a handle on the type of diet that our ancestors had and their predisposition to disease as a result of that. But what I wanted to say was that quorum sensing is a very old thing, because quorum sensing, when the bugs were alive in those coprolites, will have been going on. R is for reporting. These are newspaper headlines about the microbiome from the last few months, and you can see for yourself the sort of things that are being said there. It's a little bit more positive than when we had the legislative picture dominating things, but you can see there's a lot of things going on. I think one of my favorite one is that one, the microbiome goes viral. But those are books which are now available, or lay books on the microbiome, which, you know, I thought when I first started researching this area, I'd never thought I'd see glossy picture, nicely presented books, which for the lay public, which looked at human gut microbiome, but there's a lot of them, and there's some even just happened in the last few weeks. You can't do many microbiology talks these days without not mentioning SARS-CoV. This is the thing that's dominated our lives for the last year. Devastating effect upon health, devastating effect in terms of fatalities, but also devastating in terms of the economy, social interactions, how we behave and how we will behave in future. Just bear in mind again the study I mentioned about the respiratory issues with probiotics. So we have data, and that wasn't the only trial, because there's about 20 where probiotics can help reduce the incidence and severity of respiratory problems. So when SARS became an issue, of course, or COVID became an issue, I wrote a blog about a year ago suggesting that maybe it might be worth thinking about pre and probiotics and boosting gut health, and concomitant issues upon immune function might help in this regard. And that then led us later, towards the summer of last year, to write to the Secretary of State for Health in the United Kingdom to suggest whether, asking his experts to take a look at this to see whether there was any link between the gut and COVID-19. And we didn't get, well, I'll show you what reply we got in a minute or two, but since then, numerous research papers have begun to come out, making this analogy. And the idea is that the virus occurs in the gut as well as the respiratory tract. That's shown. The Chinese now use anal swabs as a route of detection. Sewage is used as a kind of a track and trace to try and look at population outbreaks. The receptor site in the gut and the respiratory is the same. It's called ACE2. We know that gut microbes can dampen inflammation, and one of the things about COVID is it causes inflammation. We know that gut bacteria can inhibit viruses and can stimulate the immune response. And some of these research papers which have appeared now have suggested that people who have a more compromised gut microbiome have bigger issues over COVID. And at the bottom right is the link to the letter that we wrote to the Health Secretary, which was signed by 125 scientists in the field in the UK. The response we got is summarized in that slide. We didn't get any response whatsoever, which was disappointing. But in a way, we are glad we did the letter, because other countries in the world did see what happened and took the same kind of approach with their governments, and that was in Holland, the Netherlands, Finland, Japan, China, and France. However, the story moved on a little bit further with the first trials, which actually used probiotics as a way of actually modulating the COVID symptomology. And this was a result from the first one of these, which came from Italy. Basically, people given probiotics had less referral to ICU, less fatalities, etc. And since then, there's been further studies kind of showing the same thing. So this data from Kiel on the influenza does have relevance for COVID-19. Now, the way out of this is obviously vaccination, the big vaccination rollout that's happening. But what I would like to say as well is that pro and prebiotics have been researched as adjuvants to vaccines, so can maybe help them do a little bit better. And this is my final word on COVID-19, and this is our response back now to the Secretary of State for Health and Social Care. It's a series of raspberries. There we are. T is for therapeutic modulation. This is ways in which you can modify your gut microbiome to improve health, like fecal transplants are now being used, drugging the microbiome, synthetic biology. But on the right there in the orange sector is the one I'm talking about, dietary modification. U is for US Army. We now do research with the US Army. That's a famous quote: dysentery has been more fatal to armies than powder and shot. And what we're doing here is to put prebiotics into ration packs. On the left, you've got a picture of the contents of a ration pack there, to improve gastrointestinal as well as cognitive health in the military. V is for vagus nerve. The vagus nerve connects the gut and the brain. This is where the neurotransmitters manufactured in the gut microbiota are thought to be processed at the level of the brain. Both positive and negative entities occur, and that's, you know, it's a very exciting area. It's a new development in the gut microbiome, but if we can begin to modulate gut bacteria to produce more beneficial neurotransmitters, we could be in good shape moving forward in that side of the field. W is for wind. One antisocial consequence of having these bacteria is that they produce gas. They produce lots of wind. The picture there in the photograph is a disorder called pneumatosis, which is a disorder of gas metabolism. Basically, fermentation in the gut comes from largely food ingredients as well as some endogenous sources, and produces three main groups of metabolites: organic acids, nitrogenous compounds, and then in the middle, gases. And that was a survey from a United Kingdom newspaper which showed that bloating was one of the main health issues which affected people in the UK. X is a little bit of a cheat. I've called X syndrome X. This is what metabolic syndrome used to be called. The area of gut microbiome and obesity, risk of metabolic syndrome is again another hot area. Our attempts with prebiotics are shown in the bar chart, where we gave a prebiotic GOS in yellow to people who were at risk of metabolic syndrome. and we managed to increase bifidobacteria. We managed to lower markers of insulin resistance as well as blood lipids, but did not see any effect upon weight. So that's an early trial which looked at modulating the gut microbiome of obese people, and we're looking towards doing more longer term studies to see whether weight can be more effectively managed in that regard. Y is for peptide YY or Y peptide. It's thought again that the microbiome can affect hormones and peptides which are involved in appetite regulation and satiety, and one of those is called PYY, and that was my choice for Y. So the interaction of the gut microbiome with peptides is the key there. And then the last slide is Z, and this was picked really nice. Bob Rastel, who works with Naranjan and me, is really great at doing good slides, and he gives me a lot of these. And this is one of Bob's slides here, and this is a view of what's going on inside the gut. You've got immune regulation, prebiotics fortifying positive microbes, reducing pathogens, producing acids which may help mineral sequestration, something I didn't talk about. And then you've got the whole area of appetite regulation here. But it's also suggested that some of the products of the microbiome can influence things like membrane, gut membrane integrity and tight junction ratios. And one of the metabolites, one of the peptides which is influenced in that regard is something called zonulin. And that was the choice there for Z. So that's the A to Z, and that's me finished now. If you want to ask me any questions, I'll have a look at the chat now, and please feel free to shout up. And I'd like to thank you very much for your attention. Thank you.
Thank you so much, Professor Gibson, for this interesting talk. I thank you, all of you, also for participating, and we'll open up the microphones if anyone want to ask a particular question to Dr. Gibson, or if not, you can write them on the chat and I can read them out loud for you.
Oh, I like Li Lang. She's a very nice person. Shall I answer that? Or shall I answer Jonas first? Okay, Jonas. So pre and probiotics are both used to try and prevent bloating. The target microbes for pre and probiotics are usually lactobacilli and bifidobacteria, and they cannot make gas. So if you have gas being produced inside the large intestine, which obviously does occur, it's not from those groups. So if you displace the gut microbiome more towards lactobacillus and bifidobacteria, you should get less gas being formed. And the studies that we've done in, let me get this right, in elderly people, IBS, and another one which has escaped me for now, or healthy people, all showed reductions in bloating when the prebiotic was used. So Li Lang has said there are many papers describing new strains of probiotics, value for more strains. Well, there are, you're right, and the probiotic area is pretty confused because, as I said, there are some very good products out there and some which are less robust. And also the field is, as mentioned, dominated by lactobacilli and bifidobacteria, but they don't do everything. They make lactate and acetate as part of their metabolism, but other desirable metabolites are things like butyrate and propionate. So in the future, we may have to shift our attention towards other microbes, which are not just the traditional ones, if we want to see things like propionate, which is an appetite regulator, and butyrate, which is a fuel for colonocyte function being increased. So does that answer your question? Many of the good probiotics don't eventually make. You're right again about probiotics maybe don't make it into commercial use. The study we did right at the start of our work on autism years ago was a non-commercial product, and it worked very well. And of course, what we would have hoped to do would be to recommend that product, but because it was non-commercial, we couldn't. So there has to be a bit of a balancing act to keep the research as independent as it ought to be, publish bad as well as good data, keep it, in a way that can be available as well. So you want products which people can actually get their hands on, but also it has to be as independent as possible, of course. Otherwise, it calls into question the integrity, and we can't have that. Okay.
I have a couple of questions. Well, before this one, I would like to ask a very general one. What kind, because we for sure must eat our research, what kind of probiotics and prebiotics would you recommend us? Like commercial, if it's better to consume them in a yogurt or kefir, or if it's better to buy them, I don't know, in pills. And what kind of fiber or dietary fiber should we be consuming, particular isolated one or from fruits and vegetables and cereals? Or...
Great, thank you. So the prebiotics first, the more widely researched ones are FOS, which is fructooligosaccharides, and GOS, which is galactooligosaccharides. FOS is also called inulin, and that's in a lot of plants, fruit and veg, banana, garlic, asparagus, artichoke, onion, chicory, for examples. GOS is manufactured, that's synthesized, but they're both pretty good prebiotics, and they're being used now as supplements, but they're also being incorporated into foods where the existing prebiotic level is quite low. For instance, prebiotics now being put into baked products like breads, infant formula is very popular vehicle, even biscuits and things like that are being used, cereals as well. For probiotics, you can get them in drinks like and yogurts. Dairy sector is popular, but also you can get them as freeze and spray dried products. So you get these as powders, capsules, tablets, etc. People like, I think, the drinks because they're pretty easy to take. You know, you can take one at breakfast time and use it like that. But you can't get probiotics in products that are heated because the heating would kill them. But the advantage, I guess, of the dried products is maybe you get more of a higher dose in those particular products. But again, it's difficult because there's so many out there, and it's unless we get good communication patterns and legislative procedures which allow the best research ones to actually, you know, publicize that in an objective manner, it's going to be a very confused picture. I myself take 2 a day. I take a drink and I take a freeze-dried version as well.
Thank you so much. I'll translate this next question for you from Veronica Reyes. It says, what's your opinion about antioxidant dietary fiber?
Yes, that's a good one. Again, there's a discrimination between, well, prebiotics were kind of generated because of probiotics, but because they are fermentable substrates, they've been locked into the whole fiber kind of concept. And very much, it used to be the case that prebiotics grew bifidobacteria and that happened in the gut, but the concept has been expanded recently into other areas that have a microbiome, like the skin, the oral cavity, etc. And together with that, the types of prebiotic candidates have also been expanded into things like polyphenols, antioxidants. So as we move forward, the dual effects of prebiotics may become more relevant, such as an antioxidant activity together with a typical fiber type prebiotic activity. I think that's something which is going to gain momentum, but of course, they have to be proven in vivo. And one of the criteria for prebiotics is that their efficacy should be tested in populations where their use is geared towards. So for humans, that's a human trial. For cats, that's a cat's trial, dogs, etc. So they must be confirmed in vivo. I think we're beyond the phase where we can grow microbes in a lab and claim prebiotic effects without any in vivo testing. So, but in vivo studies, particularly humans, they're expensive, they're quite difficult to manage. So we need to really do a range of preclinical testing, if you want to call it that, to give us the best chance of some degree of success in the in vivo study. You wouldn't kind of do one blinded, you do maybe some screening in lab studies to get to preferred candidates. Okay, so where are we?
I don't know if you have time for 5 more questions that we have in here.
No worries. Yeah,
perfect.
Yeah, no worries.
Do you want me to read them or do you prefer them to?
I don't mind. So what we got from Maria Esther, have you observed an improvement at ease from gastritis with probiotics? I haven't seen that, Maria. I wouldn't be surprised because well, gastritis, but gastroenteritis is something which probiotics do. They don't really have massive effects in the stomach because the transit time is too quick, and obviously it's acidic. So if you're specifically talking about the gastric area, I guess the chances are relatively low. I think mainly pro and prebiotics have their influence in the lower bowel. So another Maria, in terms of diagnostic analyses, is there a fast and accurate way to detect the flora? Well, yeah, there is. There's companies now set up who can analyze the gut microbiome. I'm not a great fan of this because there are issues over how the techniques are applied, the transport, getting them there, and interpretation of the data afterwards. I think if we want to do this in a more robust manner, which is probably not too realistic, unfortunately, I think we need to set up fermentation models and look at these over a period of time. But logistically, of course, that is difficult. I mean, there's been suggestions where ultimately we might all have a kind of a bar chart of our gut microbiome, but of course that's going to change because diet is a big driver to the microbiome. So if we change our diets markedly on a day-to-day basis, our gut microbiome is going to respond variably to that. So do you think probiotic metabolites, postbiotics? Yeah, okay. So postbiotics are the metabolites produced by probiotics, or I think they're suggested to be particles of probiotics as well. Again, this is not as advanced a research area as pre and probiotics. I see things happening and I see things being talked about in this regard, but I wonder whether if a pre and a probiotic do their job properly, those metabolites should be manufactured in the gut, and therefore is there an authentic role for postbiotics in addition? I don't know, but that would be my, I guess, devil's advocate type answer to that one. Metagenomics and metatranscriptomics approaches have an important role. Well, metagenomics certainly has, because we can characterize a gut microbiome now without inoculating any petri dishes. Metatranscriptomics is an interesting one, and just yesterday I had a meeting with Bob and myself, one of our PhD students, who was just saying that in the future metatranscriptomics is going to be a really important area for the field, because then we can understand the functionality of the microbiome. At this stage, I don't think it's having a massive impact, but as clever people begin to develop these techniques and the throughput becomes quicker, I would hope to see that kind of application. So metagenomics, I think, yes, because they've produced, the molecular biologists produce good tools to accurately monitor the gut microbiome. Metatranscriptomics, no, but could have an even bigger impact in the future than metagenomics. Okay. I think that looks like it. Okay.
I have one. I have one myself.
All right, great, yeah.
A couple of years ago, we tried during my postdoc with with a couple of professors, also from the food engineering area. We were trying to kind of model mathematically the behavior of, like in a general way, as a huge bioreactor of precisely the microbiota based on engineering equations, right? Like mass transfer, heat transfer. Would you say, is that possible? I mean, it's way too complex. And then when we started all of them, yeah, the interactions among them, and it ended up being kind of complicated. So would you say it's possible? Because I've seen the model that, well, the equipment that you used is basically a reactor, right? So would you say it's possible just to save us a bit of time, or is it way much better to go straight forward the human studies?
And I think we need better models. Our models are pretty good microbiologically, but they're not the real gut. Anatomically, they're nowhere near, and of course, we've not got immune function, we've not got absorption. So it would be great to have some clever engineers like yourselves look at these and see whether there's any interest in, for instance, having biofilms in there, having dialysis to mimic absorption, as you said, mass transfer balance would be great to have in there. So I think there are various ways in which we can improve our rather crude models which we have, which you've correctly spotted, are really just straightforward. But they're based on the chemostat principle. They're not a chemostat because they're multiple stage, but they're continuous culture. So they're not very complicated at all. They're pretty easy to build and certainly can be improved. But I myself don't have the engineering capacity to get to grips with that. So if anybody has any interest in potentially working together on that, I'd be delighted.
Take me with you, but don't tell my boss.
Okay.
Okay.
Switch off the record.
Thank you so much, Professor
No
Gibson. I don't know if anyone have a last question for our speaker. I don't think then we have anything else. So again, thank you so much, Professor Gibson, for this interesting, very interesting talk. And thank you all of you for participating. Keep tuned, please, and check our website page, our Facebook and Instagram and LinkedIn for more information regarding our next presentation. And again, thank you so much. Gracias, Dr. Gibson. Thank you so much. And thank you, Dr. Niranjan. I think he was somewhere in here. Dr. Well, I think he's here, but I'm not sure. Anyways, well, thank you so much and see you soon. Bye.
Sorry. Yeah,
Oh, here you are. No, I just wanted to say hello.